reflectometry measurements for the lipid bilayer actually fall basically on top of the VDAC protein, so you get scattering from the lipids and the protein, sort of suggestive of this model of the protein being basically embedded within a bilayer. And so this is in the process of now being further validated that this has worked, but it again demonstrates the possibility to first generate these surfaces by immobilizing protein and then backfilling with lipids. And the power of the neutrons, again, to identify those different components selectively to say where we are. And the last example I wanted to show from Hirsch's group effort is a collaboration also with scientists at IVDR, is using this idea of functionalizing the surface and then trying to ligate domains together. So the idea is many membrane proteins are very, very hard to express, but the soluble ectodomains of these proteins, like for many receptors, can be expressed in very large quantities, and there's lots of crystal structures on these domains, but they're all anchored in the membrane, many with single transmembrane helices. So the idea would be, could one actually insert these transmembrane regions of the protein first, and then link up the remaining part of the protein. So to first demonstrate this, this slide shows that one can actually indeed functionalize the surface with these peptides, and then using a peptide that's ended with a streptavidin binding moiety, you can bind the streptavidin to the top, and you can show that through, again, SPR response, that you can actually get this kind of a reassembly of a transmembrane or transhelical anchored streptavidin using these kinds of approaches. And again, the idea down the road is to basically use those same kind of